I guess that's good.
How's it going, everybody?
Good. How are you doing?
I'm doing all right. Seems like it's been a long week in spite of the fact that we didn't meet with you on Wednesday. Daniel, did you get a haircut?
Yes, I did.
Looks good. Looks good.
Thank you. Thank you.
I got a haircut also.
My 11:15 class got canceled, so I was like, might as well.
Oh, so what are protocols at barbershops nowadays?
Well, I mean, this one's really relaxed, because it's only 2, maybe 3 people working there right now.
Uh-huh.
It's called Brandles or Brundles. It's just right down Mill Street.
Oh yeah. Don't be getting the COVID though, just to get your haircut.
Yeah, I wore my mask.
Okay, good.
Yeah.
Because you guys got the email, right?
Yeah.
Yeah. Yeah. I've never been so glad that I cut my own hair. Do you guys watch late night humorists like Stephen Colbert, any of those guys?
Yeah.
Oh yeah, man, they're all letting their hair grow out.
Yeah. Have you seen Trevor Noah?
Oh yeah, he looks very different now than he used to. Yeah. Oh, Cameron, there was an email from the provost basically saying that we are moving to COVID alert level C, which basically means that all classes are supposed to go online. So for the rest of today, and I think it said through Monday, all classes should be online. And I think we'll probably get an update on Monday as to whether that will continue into the future. So
Yeah, I'm going to email and get one. So.
Oh yeah, not a big deal. I just wanted to even if Daniel hadn't gotten a haircut, we would have called attention to it today in class. So don't plan on going to any other classes, in-person, face-to-face classes today or Monday, and we'll look for an update. This is one of the reasons that we opted to go online synchronous for this class, is because we suspected that there would come a time when everybody would be online anyway, and everybody's going to be online after Thanksgiving. So why pretend and risk people getting sick when we could just kind of, like I've told Dr. Judd and other people, 100% chance of inconvenience versus a non-zero probability of somebody getting sick and maybe seriously ill and maybe even dying. So here we find ourselves. It took us longer to get to this point than I thought it was going to. My money was the end of September, beginning of October. So we've done a better job than I thought we would, and I'm pleased about that. So
yeah, I thought we were going to be here for 3 weeks.
yeah, easily. And hopefully, you know, by tightening things down right now, this little flare up will take care of it and get back to normal, whatever normal is now. But we'll just have to wait and see. So keep an eye on your emails and look for updates from the administration on what next week is going to look like. If they prevent us from doing in-person work next week, we'll just modify lab to accommodate that.
Dr. Judd,
Yeah.
where I was not able to find the PowerPoint from last lecture.
It should be up, but let me see if I so I'm pretty sure that I just changed the it should say October 30th, November 2nd photosynthesis lecture, because this has taken a little bit longer than I expected to get through all of this via Zoom. But let me see if I can just confirm that it's up there. Yeah, so under the week of November 1st to November 7th, it should say 2 November photosynthesis. That should be the same stuff that we are going to wrap up today.
Oh,
We
okay,
started last time.
gotcha. All right, thank you.
Okay, let's see if we can see if this first slide that we're going to start with looks familiar. Okay, as I said, as I just mentioned to Daniel, this is the goal was to get through C4 photosynthesis on Monday, but I'm going over a little bit. So we'll wrap this up with C4 and a couple sort of thought experiments, and over to Dr. Lindsay after that, and we'll see how far we started in just a moment. Okay. Screen. So when we wrapped up last time, I told you that the enzyme that mediates the carbon fixation step in photosynthesis, called Rubisco, indicated by this oval here, is not perfect at fixing carbon dioxide to that, that 5-carbon RUBP molecule. Sometimes it slips up and it fixes oxygen to RUBP. Now, it does pretty good at getting CO2, but having this sort of slop built into the system creates a problem when the amount of CO2 inside the leaf, that CI value, that internal CO2 value, as that goes down, as the plant is using up the CO2, the risk of switching over into this yellow cycle, the photorespiration cycle, becomes greater and greater. And the drawdown of CO2 in the leaf can happen specifically when the plant is water stressed, closes the stomata, so it's not exchanging oxygen and water for CO2, and it draws down that CO2 through the Calvin cycle, and then it starts to use this photorespiration cycle, capturing those O2 molecules. So that causes a problem, because notice what happens in this photorespiration cycle. Instead of getting 2 3-carbon molecules, you get a 3-carbon molecule and a 2-carbon molecule. It's 5 carbons. That's the same number of carbons as in RUBP, because you're not getting that extra 6th one from an oxygen molecule. So how do you keep the system alive so you can get back to the Calvin cycle? Well, the plant uses some energy, releases some CO2, creates these PGA molecules, uses some more ATP to rebuild the RUBP, and it's ready to use CO2 next time around. So this is an energy, this is a costly process that the plant has to expend energy to restore the RUBP so that you can successfully use the Calvin cycle and generate sugar, generate the products of photosynthesis. So what you need to know is the risk of photorespiration goes up when the concentration of CO2 goes down, and that happens primarily when the stomata have to close to conserve water. I think we ended with this diagram showing that when inside the leaf, you end up in a low CO2 environment and a high, especially a high temperature environment. That's where you're going to have a lot of water use, water stress. That's when the risk of photorespiration becomes very high. So this y-axis here where we're red. We've got high risk of being in that yellow photorespiration cycle when you've got low CO2 in the chloroplasts and you've got high temperatures. The solution to this that certain plants have come up with is called C4 photosynthesis. Here's a figure I've added. This one does not appear in your in the slides that are posted. But what I wanted you to see is this three-dimensional model of the Rubisco enzyme. Notice its shape, it's got structure, it's got what we call primary, secondary, and tertiary structure. And there are pockets, the pocket is indicated by the red right here, where that 5-carbon RUBP molecule is awaiting a carbon dioxide or a CO2 to initiate that chemical reaction in the first step of the Calvin cycle. So I just wanted you to have kind of a mental picture of what this RUBP looks like in relation to the Rubisco enzyme, because it's usually just drawn as a cartoon or a little diagram like this, the little 4-lobed Rubisco cartoon. But it's a protein, an enzymatic protein with a complex structure, and a little pocket where the actual carbon fixation reaction happens. All right, so the solution I said was C4 photosynthesis. C4 photosynthesis might seem rather complicated, but basically you're just adding an extra step to the C3. This image on the top left is a cross section of a leaf. And what I want you to see is that there's a lower epidermal layer down here with these kind of flattened cells. That's the bottom of the leaf, the underside. And there's an upper epidermal layer up here. That's the top of the leaf. And then in between, this is all of the thickness of a leaf, right? So just a millimeter or two thick. There's several different kinds of tissues. There's a spongy layer where there's some gas exchange that can happen, where the carbon dioxide can get from the atmosphere into the cells, and when water can get into the cells. There's an upper level here that's kind of dense and vertical, kind of stripy. That's called the palisade mesophyll. And those cells are chock full of chloroplasts. That's where a lot of the light reactions happen. They're full of chloroplasts, and when it's shady, they all kind of spread out. And when it's really, really sunny, they'll kind of stand single file in a vertical row and kind of shade each other so they don't get burned out. They don't get too much incoming radiation. And then this kind of reddish squiggles, those are the veins. If you hold up a leaf to the light, you can see the pathway of the veins that are delivering water throughout the leaf. So that's what you're catching right here. And a typical mesophyll cell, whether it's palisade or spongy, has the capacity to run the Calvin cycle. CO2 and RUBP are fixed by the enzyme Rubisco. You make the 3-carbon molecule, then you make the sugar, and then you regenerate the RUBP. That's the Calvin cycle. In a C4 plant, this Calvin cycle is restricted to only a few cells that surround the veins. And they're modified, they're enlarged. We call these bundle sheath cells. So the Calvin cycle happens in here. And in the remaining mesophyll, we have an extra step for carbon fixation. Instead of using Rubisco to fix carbon dioxide, C4 plants use a different enzyme called PEP carboxylase, or PEP, you often see it abbreviated as PEP-C. PEP-C isn't sloppy like Rubisco is. It only grabs, or only fixes carbon dioxide to this organic carbon molecule here, phosphophenol, and it makes a C4 acid. So the addition here, the extra step, is instead of using Rubisco, which is kind of sloppy and we'll use oxygen sometimes, we're using an enzyme that only ever fixes carbon dioxide. So the risk of using oxygen, the risk of photorespiration goes way down. Once this C4 acid is created through carbon fixation, it's modified into what we call malic acid and pumped into the bundle sheath cells. So moving these C4 molecules into the bundle sheath cells takes a little bit of energy. There's a cost there. But the benefit is you don't have the risk of photorespiration. Pep C in the mesophyll cells out here never uses oxygen. Once you get into the bundle sheath cells, this C4 acid, the malic acid, is decarboxylated, releases CO2 into the bundle sheath cells in an area that you can think of as being oxygen-free, so no risk of photorespiration. Rubisco is only expressed, it only occurs in the bundle sheath cells, and the Calvin cycle proceeds as it would in a C3 plant. So it's a spatial separation of the carbon fixation step and the Calvin cycle. Whereas in the C3, the whole thing, carbon fixation and the remainder of the Calvin cycle, can happen throughout the leaf mesophyll. Here's a cartoon version showing those bundle sheath cells. The gray here is the leaf mesophyll, and then the green is the bundle, the inner green layer is the bundle sheath. And it's called a bundle sheath because the vein, we often refer to veins as bundles. So it's sheathing the vascular bundle or the vein. That's why we call it a bundle sheath cell. So that's these right here. This is the only place where Rubisco, the enzyme that starts the Calvin cycle, is expressed in high concentrations. Out here, it's only Pep C. So you can, so you don't have that risk of photorespiration. If you were to take a leaf and look at it from above, this is, I think this is a corn leaf actually. These kind of railroad looking areas are the veins. So that's where water and sugar is being transported. The cells you're looking at are xylem cells, and those are the ones that, that transport water, and they have these thickenings, these bar-like thickenings. Then these fat cells around the veins that surround them here and over here on this big vein, those are the bundle sheath cells. And then the thinner, kind of whiter, kind of washed out cells, those are the typical mesophyll cells where Pep C is expressed. And these are the ones where only Rubisco is expressed. So Pep C, carbon fixation, Rubisco to start the Calvin cycle. I put together this slide for those of you who wanted a little bit more text on some of the slides for how bundle sheath cells work. So this is something you can refer back to, but what I'd like to do is show you a diagram of it and walk you through that, and then you can refer to this. So away from the veins, in the typical mesophyll cell of a C4 plant like a grass, CO2 comes in, and this enzyme represented by the blue hexagon, Pep C, fixes the carbon dioxide and makes this oxaloacetate molecule. It's a 4-carbon chain. So 3 plus 1 equals 4-carbon chain. And then NADPH, remember that's the energy carrying molecule, is kind of spent on converting this OAA into malate and pumping it into the bundle sheath cell. Once it gets into the bundle sheath cell, the carbon dioxide is released. There's no oxygen in here, so no risk, or very little, so no risk of photorespiration, and the Calvin cycle proceeds. You get that sugar, and the sugar can be loaded directly into the vein because it's right next to the vein. Meanwhile, the rest of the carbon molecules or carbon atoms that are not released as CO2 can be exported back out to the regular mesophyll where carbon fixation can happen. So carbon fixation or C4 cycle, and then the C3 cycle is the actual Calvin cycle that uses Rubisco. So it's really all about counting your carbons, right? When we did the Calvin cycle, it was 5 plus 1 equals 6, makes 2 3-carbon molecules. You can combine 2 3-carbon molecules to make a 6-carbon molecule called a sugar. And then the rest of them are used to regenerate this RUPP and keep the cycle going. So what are the benefits? Maybe some of these are predictable at this point, but because Pep C doesn't react with oxygen, if the plant has to conserve water and close its stomata, the carbon dioxide concentration in the leaf goes down, 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 but even at those very low levels of carbon dioxide in the leaf, Pep C can continue to fix carbon dioxide, create those malic acid molecules, and pump them into the bundle sheath cells. They're also a little bit more efficient when it comes to building Rubisco molecules. or building Rubisco enzymes. I told you before that Rubisco is one of the most abundant enzymes on the planet, if not the most abundant. That's because it's not very efficient, partly because it's kind of sloppy and will use those oxygen molecules. So by concentrating the Rubisco in the bundle sheath cells, you don't need to express it throughout all the leaf tissues. So you don't need to use as much nitrogen to make those enzymes, and the plant can be a little bit more efficient with its nitrogen. So they get more efficient with their water because they can close their stomata and keep producing sugars, and they're also more efficient with their nitrogen because they only have to produce Rubisco in those bundle sheath cells around the veins. These are really good benefits. The trade-off, the cost, is that it requires energy to make those 4-carbon molecules like malic acid and pump them into the bundle sheath cells. C3 photosynthesis, the kind we've already talked about, has been operating on Earth, it's been operating on land for about 400 million years, and it's been operating in algae in the ocean and in freshwater bodies for even longer. C4 photosynthesis is a kind of a recent invention for plants. It's only been around since roughly the last 30 million years, maybe a little bit more. But it's become more common in the last 8 to 10 million years. And we can think about why. When you have high temperatures and low CO2, those conditions favor C4 plants. Let's think about temperature first. If you've got high temperatures, water evaporates quickly. We talk about the atmosphere as having a high evaporative demand. The atmosphere sucks water out of the soil, it sucks water out of the plants, the water evaporates or transpires through the plants. When that happens, the plants are at risk of wilting, of drying out. So they have to close their stomata and conserve water. If their stomata are closed, they can't take in CO2, and so the risk of photorespiration goes up. And C4 plants get away with that, right? They don't have to worry about photorespiration because they're using Pep-C instead of Rubisco to fix carbon. Similarly, when the stomata are closed, the concentration of CO2 in the leaf goes down. Well, it turns out that the amount of CO2 in the atmosphere has also changed through time. And the ability for plants to take CO2 in depends on the concentration of CO2 in the atmosphere. It's all based on diffusion. So when you have high CO2 in the atmosphere, that's good for C3 plants. But when you have low CO2 in the atmosphere, then they operate at higher risk of photorespiration. C4 plants, by contrast, since they're already fine at dealing with low CO2 in the leaf, they can handle low CO2 in the atmosphere as well. So when you have low CO2 in the atmosphere, like we do now, around 400 ppm, and you've got high temperatures, like in the south or in the tropics, then you find C4 plants are favored. When you have low CO2 and it's cool, then C3 plants are favored. And throughout much of our geologic history, we've had much higher concentrations of carbon dioxide in the atmosphere. So we're in this region where even if it's warmer, you're still in the C3 favored region. It's only in the last 20 to 30 million years that CO2 concentrations in the atmosphere have been going down, down, and down to create the world we live in today. And during that time, while CO2 has been declining, C4 plants have become more and more abundant. This graph shows the sort of the advantage of being C3 or C4 under different CO2 concentrations. So notice that, well, this figure is a little bit old. Today, our atmospheric CO2 levels are at about 400 ppm. If you go up here and read over to the y-axis, notice that the photosynthetic rate is a little bit higher for C4 than C3 plants on average at 400 ppm. So C4 plants are favored when in reasonably warm temperatures at current levels. As CO2 concentrations go up, the lines cross and you'll have situations where C3 plants are favored. But it's pretty common to be down here right now where C4 plants are favored, especially at high temperatures. First, I want you to turn your attention to the graph over here on the left. This is a graph showing the relationship between leaf temperature and photosynthetic rate, or the rate at which the plants are fixing carbon out of the atmosphere. And this graph is plotting the relationships under bright conditions. So think middle of the day. At low temperatures, when it's cool, the C3 photosynthetic plants have a higher rate of photosynthesis. And the C4 plants have a lower rate. You can see the dashed line is below. But as temperature goes up, the lines cross and C4 plants gain an advantage. And notice they gain a really big advantage. So once you get up here at about 30 degrees centigrade, which is pretty warm, the photosynthetic rate of a C4 plant is much higher than that of a C3 plant. In fact, the C3 plants start to decline. If you think about that over the course of the whole, or spatially over the course of a map, where do we experience the warmest temperatures on Earth? When you think of hot climates, what do you think of?
The equator.
Yeah, you think of the tropics, right? Low latitudes. When you think of a tropical rainforest, what kind of vegetation do you imagine?
Like high canopy, big trees.
Yeah, you think of large canopy, you think tall trees, you think vines in the rainforest, right? Those are in environments that are warm, but they're also wet. There's also plenty of water there. And pretty much all trees are C3 plants. So if you look in the Amazon here, or the Central African rainforest here, or on the islands of Southeast Asia, that's where we have rainforests today, you see a lot of white and a lot of purple. So you're down here in a low proportion of C4 plants because these are forests. But as you start to get into tropical savannas and deserts, the proportion of plants that are C4 goes really high. So we're up here in the yellows and oranges and reds. Most of the plants, most of the grasses in tropical savannas, and as you start to get into deserts, are C4. In real deserts, we get cactuses and euphorbs and things like that, succulent plants, and it's a little bit different. So don't worry too much about the Sahara Desert here. But in these red areas, you're looking at tropical savannas, where there's a lot of grasses. And then as you get into slightly higher latitudes down here in South America and into the southern plains of the United States, we see we're in these green and blue regions. This is where we have grasslands like our prairies, and there's a lot of C4 plants, especially in the southern plains. Notice the red to green transition here from Mexico and Texas and this region of the US. And then as you get into higher latitudes, up here like in southern Canada, throughout the steppe region of Eurasia here, then we get into grasslands where the grasses are mostly C3. We've given you examples of C3 and C4 grasses in our plants of the day. I want you to think that when you hear C4 grasses, think tropical savannas and the southern plains, the southern prairie. If we zoom in on North America, we can really see the way climate structures the kinds of prairies that you can find. In the east, we have our tall grass prairies. And in the west, we have our short grass prairies. So tall grass C3 is this yellow color, and tall grass C4 is the green, dark green color. This is the Kanza Prairie region right here. And then the blue and the light green are short grass. So short grass should make you think drier climate. Tall grass makes you think wetter climate. And then warmer climates in the south C4 plants, C4 grasses, cooler climates in the north, C3 grasses. So you can really get it, if you get to the point where you can recognize tall grass and short grass prairie and you know your C3 grasses and your C4 grasses, I could give you an example community like well, we can think about the ones at Konza Prairie, tall grasses that are C4 like Andropogon and Sorghastrum nutans. That will tell you, okay, tall grass C4 plants were probably in the southeastern portion of the prairies in North America.
Hey Nathan,
Question.
before you leave that,
Yeah.
notice down there in kind of north to central Texas, you see that there's some more C3 grasses, C3 tall grass prairies. It's warmer down there, but as you move closer and closer to the Gulf of Mexico, it also is getting wetter. And so you also have to consider the proximity to the moisture blowing in from the Gulf.
Yeah, there's, I think we had a question, but that reminds me, I wanted to point out that when it comes to water availability for plants, you have to think about what's available in the soil, how much water is getting into the soil from rainfall, which is going to be high as you get close to the Gulf, and also how humid is the atmosphere. If the atmosphere is not very humid, the plants have to lose a lot of water when their stomata are open just to gain a little bit of carbon. But when it's really humid, then the evaporative demand of the atmosphere is not as great. And so the plants can get a little bit more carbon for a certain amount of water that they lose. So there's two aspects of water, atmosphere and soil.
Yeah, I worked at a tall grass prairie when I was in grad school north of Dallas, and it was very humid and kind of miserable to work in. And the water holding capacity of the soils was also very high, such that at the tops of hills on this prairie, there were actually crawdad burrows because the heavy clay content of the soils held water so well.
We had a question?
Yeah, so this is backtracking a little bit, but with C4, okay, I guess I'm confused. So C4, it's like disadvantages that it uses energy, right?
Yes.
Okay, and then
Sorry.
if we don't have C4, then we can go into cellular respiration, right?
It's photorespiration.
Photorespiration.
Remember,
And
this is all happening in the chloroplast, not the mitochondria.
okay, but photorespiration, that still uses ATP, right? Is it just that C4 uses more?
So both of them are costly, right? So if you're spending ATP, let me stop sharing for a second. Yeah. This is a good point to come back to, Tessa. So for photorespiration, the main goal of the plant is to reconstruct the RUBP molecules so that you can get back to the Calvin cycle and get back to normal carbon fixation and sugar production. So it takes energy to make those 2 carbon and 3 carbon molecules back into RUBP, and in the process, some carbon dioxide is released. In the photorespiration cycle, you're taking in oxygen, Rubisco is using oxygen, and it's releasing a little bit of CO2. So in that sense, it looks kind of like cellular respiration, but it's costing energy and you're not producing any sugar, and it's happening in the chloroplast. In those ways, it's very much unlike cellular respiration, because cellular respiration is breaking down sugars, right? You break down glucose molecules to get that ATP. But cellular respiration happens in the mitochondria, and photorespiration happens in the chloroplasts. That's where the light comes from. The ATP that's used to rebuild those RUBP molecules comes from the light reactions in photosynthesis.
So it's not that they're using different amounts of ATP, it's just the payoff that they get from the amount of ATP that they're using?
Who's they? I think you're on the right track, but let me make sure.
Yeah, like the plant cells. So in like in C4, they're using ATP, but they're not getting as much payoff because they're not breaking down sugars rather than.
Oh, I see. Not quite. So in C4, you're using energy to concentrate carbon dioxide in the bundle sheath cells.
Okay.
But the advantage is you've got concentrated carbon dioxide in the bundle sheath cells, which means in those cells, the Calvin cycle can proceed and you can create sugar. So the grasses produce sugar, they grow, they do really well.
Okay.
In a C3 plant, undergoing photorespiration and regenerating RUBP is kind of a way to survive in water stress conditions, in warm or bright conditions, because you get back to this point where you have RUBP. But if it gets too hot or too dry, and you spend all your energy just on photorespiration, right? Use up this ATP here, then if the plant's not making sugar, it's gonna eventually run out of energy and die.
Right.
So spending this energy is a way to sort of survive the process of photorespiration until you get back to good conditions. Whereas for a C4 plant, you just avoid photorespiration entirely by hiding the Calvin cycle in these specialized cells around the veins called bundle sheath cells, where the rubisco never sees oxygen, and so it never undergoes photorespiration.
Oh, okay. I think I get it now.
Anybody else have a question about that distinction? I want to make sure that's clear. Sorry, me. Okay, if not, I'm going to reshare the screen. Oh, come on, Zoom. Stop doing that to me. Too many windows. Here we go. So this links us back to Kanza Prairie, right? We're in the tall grass, C4 dominated prairies of North America. And you can see where that fits into the broader picture that's kind of structured by both the biology of the plants and the climate, the two important things being water availability and temperature. So that's big picture, spatial scale stuff. Let's bring it back down to plants that you can think about because you're familiar with them. Here are two plants that we've talked about, Andropogon girardii and Salvia pitcheri. Maybe we should call that Salvia azurea. It's the same thing. But these are two plants. Who can tell me which one of these is a C4 plant? And which one is a C3?
Isn't the grass a C4?
Yeah. So some grasses are C3, some grasses are C4. Most of the tall grasses we've talked about so far are C4, and Andropogon is a recognizable classic example that you should be familiar with. And almost all forbs are C3. There's a few exceptions, especially in desert regions, but almost all forbs in the prairie are C3 plants, and Salvia pitcheri is a good example of that. So let's take a look at this graph that is comparing the rate of photosynthesis for Andropogon girardii in the black circles and Salvia pitcheri in the open circles. We also have two other C3 plants here, a goldenrod, one that you all are familiar with, and actually don't remember who the Baldwinii is, but it's another forb. And we're looking at how their rate of photosynthesis changes as the brightness of their environment increases. So we go from dark down here to very bright over here at 2000. What do you notice about the effect of brightness or of light availability on the rate of photosynthesis for C4 versus C3 plants? Somebody take a stab at explaining this graph. And the orange question at the top.
I think brightness favors the C4 plant, right?
That's correct. Yep. So under bright conditions, the rate of photosynthesis for the C3 plant saturates. Once you're up here, once you're above about 1,000 ppfd, you can keep increasing the brightness, but you won't really increase the rate of photosynthesis very much. With the C4 plant, as you increase brightness, you continue to increase the rate of photosynthesis. Now, this graph is generated under reasonably warm conditions in the growing season. So under warm conditions, C4 photosynthesis in full sun greatly exceeds that of C3. So this is full sun over here. Under cooler conditions, this advantage isn't as pronounced, but under warm conditions during the growing season, especially in the southern part of the US, this is a big advantage for C4 plants. So they produce a lot more biomass than C3 plants do. That's why the prairies are grass dominated. Let's think now about water use efficiency. Remember, this relates to whether the plants can keep their stomata open and have low water use efficiency, or if they close them and have high water use efficiency. So our droughts, so water stress, more likely to favor Andropogon girardii, C4, or Salvia, the C3. Let's look at the graphs here. We're looking at the C4 plants under a variety of conditions, lowland or upland, burned or unburned, and we're looking at the same variety of conditions for the C3 plant. Who has the higher water use efficiency?
C4.
Yeah. Can anyone remind me why that would be? Can anyone explain that in terms of stomata and carbon dioxide and water?
The C3 plants, their stomata closes whenever there's a drought in order to conserve water. And so they have less access to CO2. And so they can't go through the Calvin cycle as easily. And so the C4 grass doesn't have to go through that process because it's like hidden within like the C4
Yeah,
like
what were
they're-
those cells called? Someone remember?
yeah.
What those cells were called to help test it out? It's those special cells that surround the vein or the vascular bundle.
The bundle sheath cell.
Yeah, thanks, Ryan. Good. So that, as Tessa was saying, the C4, sorry, the Calvin cycle is happening in those bundle sheath cells. So Andropogon girardii can close its stomata if it needs to under hot and dry conditions, and that the PEP-C enzyme can continue to fix carbon dioxide in the leaf even as it goes to very low concentrations, and pump that carbon into the bundle sheath cells where the Calvin cycle can proceed and the plant can keep producing sugar and keep growing. So one more, nitrogen availability. Remember that nitrogen is a key component in amino acids and therefore proteins, which include enzymes like Rubisco. So if you have an environment where nitrogen is limiting in the soil, is that low nitrogen availability going to favor Andropogon girardii or Salvia pitcheri? This is a little bit of a harder one. we're looking at the amount of nitrogen per unit mass in the leaves. And you have to use that knowledge to answer this question of whether low nitrogen availability is going to favor the C4 or favor the C3. One thing that might help you as you start to think through this is go back in your mind to those Tillman diagrams, the resource ratio diagrams. The winner of those usually ended up being the plant of those competitive interactions, usually ended up being the plant that was able to keep growing with lower resource availability. So access that if you have it in your mind. And the question is low nitrogen or limiting nitrogen going to favor the C4 grass or the C3 forb? Which plant, let's do a different one. Which plant, the C3 or the C4, needs more nitrogen for the Rubisco enzyme? Tessa and Ryan reminded us that the Rubisco enzyme is concentrated in the bundle sheath cells in a C3 plant, or excuse me, in a C4, it's concentrated in the bundle sheath cells. Where do you find Rubisco in a C3 plant, if not in the bundle sheath cells?
Isn't it the mesophyll?
Yeah, throughout the mesophyll. Everywhere there's green cells in the leaf, you'll find Rubisco. So who do you think needs more nitrogen per leaf, per unit leaf? The C3 or the C4?
It's a C3.
Yeah,
Okay.
good. It's the C3. So the C4 plant can tolerate lower nitrogen levels from the soil because it doesn't need to make nearly as much Rubisco per unit leaf than the C3 plant does. Well, one way to say that is C4 plants have greater photosynthetic nitrogen use efficiency. So we were talking about water use efficiency. How much carbon do you get for the water you lose? How much photosynthesis do you get for the nitrogen that you use? They have greater nitrogen use efficiency, so they can tolerate lower nitrogen levels in the soil. So we've gone through a couple examples here where we talked about light and water. We've spent a little bit of time on photorespiration. You should be able to fill out a table kind of like this. Which plants, C3 or C4, have higher water use efficiency? Someone other than Tessa or Ryan. I know you guys are out there.
C4.
Yeah, good. C4 going to have higher water use efficiency. What about the risk of photorespiration? Who has a higher risk of photorespiration under hot and dry conditions?
C3.
Good. Yep. Because the C4, they've sequestered those Rubisco molecules, those Rubisco enzymes in the bundle sheath cells. Which one is going to have their photosynthetic rate saturate or reach its maximum first? The C3 or the C4 as you increase brightness, as you increase light intensity?
C3.
Yep, good. We had one graph showing that, and it was as you increase the brightness, the C4 was getting more and more photosynthesis, higher and higher photosynthetic rates, but the C3 is saturated. And what about temperature? Which one can photosynthesize better at higher temperatures? Which one has a higher optimal temperature for photosynthesis?
C4.
Yeah, good, C4. And there's a slide where we look at that in two ways. One of them where we saw that as you increase the temperature, who gets the advantage? And then we also saw it on the map, right? And you saw that at low latitudes in the tropical savannas, they were very much C4 dominated landscapes where you have high average temperatures. Okay, so that wraps up our discussion of C3 and C4. I wanted to show one last slide for you all because as you learn about photosynthesis and read about it in your textbook, or if you take plant biology in the future, you will also spend more time talking about a third kind of photosynthesis called CAM photosynthesis. It stands for Crassulacean acid metabolism. Crassulaceae is a family of plants. They're kind of succulent, just like we've talked about the Asteraceae. The Crassulaceae is another family, so it's named after them. Or it's named after the Crassulacean acid, if not the plants themselves. And they have an interesting situation where carbon dioxide is fixed at nighttime in the deserts. So in a cactus, the stomata open at night, CO2 comes in, they store up as much carbon as they can, and then close the stomata to protect their water. And then during the day, when the sunlight is coming, the light reactions proceed, they generate that ATP, and then they use that ATP to power the Calvin cycle using the CO2 they collected at night, that they sort of built up at night as malic acid. So the difference here is that for C4 plants, you spatially separate carbon fixation from the rest of the Calvin cycle. Right? You put the mesophyll is where you have the carbon fixation, and then the production of sugar is spatially restricted to the bundle sheath cells. In the CAM plants, you're separating it out by time. You fix the carbon at night with the stomata open, and then at daytime you close the stomata, and then photosynthesis proceeds when there's light available to power the Calvin cycle. That's the last slide I have. You might see there's a couple extras here where I show you what a C4 carbon chain molecule looks like, but this is the end here. And with that, I'll open up for any questions about C4 or CAM photosynthesis or C3. I put a link at the top while you guys are thinking about questions to a link to a Zooniverse website. This is a website where anybody can learn about stomata and help count stomata on the surface of a leaf to help us figure out how carbon dioxide concentrations have changed through time. So it's a citizen science project. Anybody can sign up. You take a little quiz about stomata and CO2 and how to count cells. And then you're given samples that need to be counted, and you can contribute to this growing database that shows how stomata in ginkgo trees have tracked carbon dioxide concentrations in the atmosphere through geologic time.
Any questions for Dr. Judd?
All right.
All right, then I'm going to go ahead and share my screen. We're going to shift gears. And I forget, Dr. Judd, I haven't been keeping as close a track as I should have with prairie grass and forb of the day. Have you done switchgrass before?
I did. We talked about that as a biofuel.
Okay, all right. We won't do switchgrass today. I'll add one at the beginning of class on Monday. Did you do prairie clover?
Nope, that's a good one.
Okay, all right. So prairie forb of the day, Dahlia purpurea, purple prairie clover. It's hard to see on this because you can't really see many of the leaves, but this ends up being a It's in the family Fabaceae. It's in the family of legumes. These legumes are the kinds of plants that fix nitrogen, and we're going to talk about nitrogen fixation probably on Monday. We won't get to it today, I don't think. So this is a C3 forb. It fixes nitrogen. It has these closed Fabaceae style flowers that are aggregated in these flowering heads. This will then develop into a little seed head. A deep taproot so that it can get access to water. Oftentimes, because this is a relatively fast growing forb, it's used in restoration. It's also used in restoration of disturbed areas because it has this ability to fix nitrogen. It's been used by Native Americans in the past as tea. And so you can take the leaves of the plant and boil them and make an herbal tea out of it. And people in the past have also used the dead above ground biomass as brooms, evidently. This is one of those forbs that develops kind of a woody nature to its stems. And so it's durable for converting into brooms. And then the prairie animal of the day is not necessarily an animal that you were looking forward to. And you haven't done this one yet, have you, Nathan?
Nope. Go for it.
This is Schizocosa ocreata. It's a small spider, legs and all. It's probably about this big. And so it's not a very large spider, but it can occur at really, really high densities. One of the easiest ways to recognize it is that it has this pale band that goes along the top of the cephalothorax, and then it has these brushes on the front legs of males. Females do not have these brushes. The reason that I bring this up at Kansa is that there's a guy over at K-State who studies food webs at Kansa, and he has found that when these things are abundant enough, they can actually alter the distribution and behavior of grasshoppers such that they can actually affect the rates of herbivory on plants by altering the movement patterns and feeding behavior of grasshoppers, actually. So we talk a lot in this class about the effect of bison, for example, on bringing about heterogeneity in the plant community because they tend to be localized feeders and they only feed on certain types of plants. But you can also have heterogeneity being added to a landscape by affecting where organisms go and forage. And that is evidently something that this particular species can do. They have a lot of little spiderlings. They carry them around in an egg sac. And then when the egg sac hatches, they climb onto the abdomen of the female and she carries them around for a couple of weeks. And then they slowly start to disperse and run off and become little spiderlings. And because of these brushes, there's been a lot of behavioral work done on mating behavior in these guys because the males use these brushes to attract females. As it turns out, males with bigger brushes have greater success with the lady spiders. So anyway, they also use these leg waving displays where they're waving their legs at other spiders as a species recognition mechanism. If you wave your legs in the right way, the female will perceive you as a potential mate rather than a potential meal, and that's always good. Questions about the forb of the day or the animal of the day? All right, so I want to back up for a minute before we start on nutrient cycling and try and frame the Kansa Prairie case study in a broader frame so you can kind of see where the pieces are fitting together. And so what we have is climate. Climate is a major driver of everything that happens in a plant community. And climate is made up of things like temperature and rainfall. And temperature and rainfall, as you've just seen from Dr. Judd's coverage of C3 and C4 plants, the temperature that you're at and how much rainfall you have available to you influences kind of what type of photosynthetic pathway is going to be most prevalent wherever you're at. These things also affect, through those photosynthetic pathways, how much above ground biomass is going to be on the landscape. So how much plant biomass is around at Konza Prairie. Now, this above ground biomass can be affected by grazing, and it can be affected by fire, which are the two main things that they look at in the Veen et al. Paper. Grazing can alter nutrient availability because of the presence of grazers on the landscape, doing things like defecating and urinating and dying. They add nutrients back to the soil, but grazing can also result in decreased biomass because the grazers are actually eating the plants. But this type of biomass loss is selective in that not all grazers eat all plants equally, and so they only eat some of the plants and leave others intact. And this is one of the things that leads to heterogeneity in the environment. Fire, on the other hand, does affect nutrient availability. In some cases, it makes the biomass easier to break down because it's burned off a lot of the things that protect the materials that are inside of the plant from making it into the soil. But then it also loses the nutrients to a certain degree because when the biomass burns up, those nutrients get volatilized and released to the atmosphere, and they're no longer available to the soil. But then fire also decreases biomass, but the decrease in biomass due to fire is very unselective. It burns down grasses, it burns down forbs, it burns down everything. And so there's a huge difference in terms of the way that fire decreases biomass versus the way grazing decreases biomass. In either case, this decreased biomass alters the light environment. How's it going to alter the light environment? Is there going to be less light or more light after grazing and a fire? Devin, more light or less light after grazing and fire? Cat. Cat Price.
I really am not sure. I'm kind of
Okay, well, if you burn all of the biomass off, is there going to be more light available to any plants that might be coming up after that fire, or less light when you think about all the plants that were there before the fire?
There'd be more light.
Yeah, there would be way more light. And so once again, we're potentially moving, if we go back and think about resource limited competition, we're moving from a community that had less light availability because there was a lot of competition for light by all the plants there, and we're moving to a high light environment because all of that plant biomass has been burned away. Same thing happens with grazing. Good. All right. So what we want to talk about over the next whatever time is remaining here today and on Monday is nutrient cycles. And so what our goals are going to be is, first of all, we need to explain, you need to be able to explain why organisms need the various nutrients that are involved in biogeochemical cycling. We're going to teach you what the pools and fluxes are for carbon and nitrogen cycles. We're not going to do sulfur and phosphorus because once you kind of grasp the basics of carbon and nitrogen, you should be able to figure out sulfur and phosphorus. We're not going to give them to you in class, and we're not going to test you over them, but the same tools that you use to understand the carbon and nitrogen cycle would be useful for sulfur and phosphorus or any other nutrient that you can think of. You need to be able to define and describe the inputs, outputs, and transformations of the carbon and nitrogen cycle. We're going to be talking a lot about symbiotic relationships among organisms that are important in the nitrogen cycle. So nitrogen fixation, what it is, how it works, how the plants benefit, and what limits nitrogen fixation. And then we'll also look at a different set of symbiotic relationships having to do with fungi in plants. And we'll talk about how those relationships benefit both the plants and the mycorrhizal fungi. So one set of symbiotic relationships with bacteria and another set of symbiotic relationships with mycorrhizal fungi. But both of these symbiotic relationships are important from the standpoint of both nutrient cycling and in terms of maintaining water balance. So it's 33 after, and I really don't want to probably get too far into this, given that we only have 2 minutes. But on the screen in front of you are a bunch of different molecules. What I would like you to do between now and Monday is look at these molecules and come in on Monday and be able to tell me which of these molecules corresponds to which of the major classes of biological macromolecules. So if you don't know what I mean by biological macromolecule, you can go and look that up. And while you're looking that up, you can also look at the chemical structures of these things and figure out which of these belongs to which class. And we'll start there. It's important to know what the biological macromolecules are and what makes them up, because the things that make them up are the things that organisms need to survive and grow and reproduce and all that. So if a particular element isn't a component of a biological macromolecule, then we don't really need to worry about it cycling in ecosystems because nobody really uses it. Questions? All right, then we'll see you on Monday. Dr. Jett and I are working on getting your literature summaries back to you. It's just slow work, and it seems like it's been a really busy week. So start looking for those coming to you over the weekend so that you can start preparing, well, hopefully continue preparing your literature summary on the Veen et al. Paper. I've already had some people in my office asking questions about it. So don't let that fall off of your calendar of things to do.
Yeah, you want to be able to use the comments from Hendry et al. To revise your drafts of your Veen et al. Summaries. So we'll get you those to you as soon as we can. We've been working on them.
All right, questions? All right, have a good weekend. Try and relax a little bit. Don't stress too much about the presidential election.